Welcome back, class. We are still on neurology, and let's look at some of the protective coverings of the brain. So, here we're seeing a cross section of the brain. We know that we have the skull, we have the bone that protects it. I mentioned earlier the meninges, and the meninges are the same meninges that surround the brain, also surround the spinal cord, and they are PAD, pia mater, arachnoid, and dura mater. Those are the meninges. And then we also have fluid that helps to protect the brain. And the fluid circulates in these chambers called ventricles. So you can see the lateral ventricle here. This is lateral ventricle 1, and this is lateral ventricle 2. Here's the third ventricle. That's 3. And then we have the fourth ventricle, right here is 4. In order for that fluid, the cerebral spinal fluid, CSF, and if you remember, the CSF is made by the ependymal cells, which is a type of neuroglial cell. So in lateral ventricles, there are two of them because one is in this hemisphere, the other is in this hemisphere. So there's two of them. And it doesn't matter whether one's on the left or two is on the right. I'm just numbering them. This could be two, this could be one. It makes no difference. There's just two lateral ventricles. And then it has to go through this interventricular foramen of Monroe to get to the third ventricle. And then going from the third ventricle into the fourth ventricle has to go through the cerebral aqueduct. Then after the fourth ventricle, it goes down this either median aperture or lateral aperture. It goes down the spinal cord, or it goes this way and recirculates around the brain and gets reabsorbed way up at the top in that superior sagittal sinus. So you have the CSF, this pump that's sometimes called the craniosacral pump. And there are lots of DOs out there and DCs out there, either doctor of osteopathy or doctors of chiropractic, that treat by working with the flow of the cerebral spinal fluid to help people with a lot of ailments. Okay, and it helps to ensure that there is healthy pumping of the cerebral spinal fluid. The pumping of the CSF is really, really important. This is why vaginal deliveries are so important in terms of birth. When a baby's coming out of the birth canal through the vaginal canal, when you look at the diameter of a baby's skull and you look at the diameter of the vaginal canal, the math just doesn't add up. Okay, so what ends up happening is the skull bones overlap, right? The skull bones are not solid. There's many sutures and fontanelles that can overlap. This is why babies look so funny and silly when they come out of the birth canal. Many of them look like they're cone heads, right? There's different shapes of the baby's head because it's squeezing through this tight vaginal canal. The bones have to overlap. They squeeze through the vaginal canal, and after the head crowns and comes out of the vaginal canal, it's like this recoiling, this springing open of the cranial bones, and it creates this pumping mechanism of the cerebral spinal fluid. Okay, it's so important, really is important. Um, in terms of blood flow to the brain, when we get into circulation, into the circulatory system, we'll go over the vertebral arteries and the carotid arteries. Those are the two main blood vessels that come off like the arch of the aorta to bring blood into the brain. Right here, that's the vertebrals. Here's another, right? You can see it here. There's the vertebral arteries. And then we have the carotid. And the carotid, as it goes up, kind of divides into an internal and external. And the internal carotid brings blood into the brain. What helps drain the brain of deoxygenated blood is going to be the jugular veins. Okay, usually for most blood vessels of the body, if there's an artery, there's a vein. So if there's a vertebral artery, there'll be a vertebral vein. If there's a subclavian artery, there'll be a subclavian vein. If there's an axillary artery, there's an axillary vein. But there is no carotid vein. What would be analogous to that would be the jugulars. The brain utilizes about 20% of the body's oxygen. And so any type of interruption of that blood flow will deprive the oxygen. And if there's decreased blood flow and decreased oxygen, there's going to be some brain damage and cell death to the brain cells. That's what we call a stroke. If it's a mini one, they call it a TIA, a transient ischemic attack. If there's glucose deficiency, right, if there's decreased flow and there's decreased glucose, then that can result in some mental confusion, some dizziness, convulsions, or they're unconscious. Why do they go unconscious? If they go unconscious, then that means they fall on the floor like this compared to standing. Gravity is working down, so it's a lot harder to get blood flow up to the brain. When you're lying down, blood flow can move in both directions much easier. So it's kind of like innate intelligence at its best. When we get into circulation, we will talk about the arterial flow, how it actually goes up the vertebral arteries, or how it can get in from the carotid arteries, and why if we get certain type of clots in the vertebrals or the carotids, we start to get decreased function in different parts of the brain. So I'm not going to hold you accountable yet for these blood vessels. When we get into the circulatory system, make sure you watch my video on the circle of Willis, because that's what this is right here. They call it the circle of Willis. The blood-brain barrier. The blood-brain barrier, remember, it's made of those astrocytes. It protects the cells from any type of harmful substance or toxin or pathogen, and it serves as a selective barrier to prevent many of the passages of substances from the blood into the brain. So this blood-brain barrier can prevent entry of certain types of therapeutic drugs. So they'll try and develop certain pharmaceuticals, and maybe they'll try and hide the drug in with high glucose. They'll try and sneak it in with large amounts of sugar because the brain does like that to function, to try and get that therapeutic drug into the brain. So here's the side view. You heard me mention cerebral spinal fluid. The blue structure, everything you see here in the center, those are the ventricles, right? So we have, from the side view, we can see the lateral ventricle. We could see one on this side and two on the other. Here's the third ventricle. And then we have the fourth ventricle here. So how does cerebral spinal fluid go from the lateral ventricles into the third ventricle? It's got to go through the interventricular foramen of Monroe. And then how does it go from the third ventricle into the fourth ventricle? It's got to go through the cerebral aqueduct. After it goes in the fourth ventricle, it's got a few options. It can sneak out through the median aperture, or it can sneak out through the lateral aperture, or it goes right down the central canal, all the way down to the sacrum. So you have this craniosacral pump pumping the CSF, there's cerebral spinal fluid. It's designed to protect the brain and the spinal cord against, chemical issues, chemical shock, or physical injuries. It carries oxygen, it carries glucose, and other substances that are important for the electrical conduction of neurons. We know it carries glucose, proteins, and ions. It's involved in mechanical protection, chemical protection, and circulation, nutrients and waste products to and from the bloodstream. In terms of chemical protection, ionic concentrations. Remember, we talked about sodium and potassium for action potentials. And in terms of mechanical protection, it helps to buoy the brain, right? It helps to buoy or float the brain to try and soften any type of impact against the the skull or the brain to avoid concussions. There's usually 3 to 5 ounces of cerebral spinal fluid, and the rate that it is being produced is the rate that it should be filtered. If you get production at a certain rate without the drainage of it at an equal rate, you start to get pressure inside the skull, and that pressure is what they call water on the brain, or hydrocephalus. Hydrocephalus. So again, there's your sagittal, your lateral view, where we can see the lateral ventricles 1 and 2. Here's where the third ventricle is, and here's the fourth ventricle. For CSF to go from the lateral ventricles into the third ventricle, it's got to pass through the interventricular foramen of Monroe. To go from the third ventricle into the fourth ventricle, it's got to pass through the cerebral aqueduct. And after the fourth ventricle, it's got a few options. It's got 3 options. You can go through the central canal, the median aperture, or the lateral aperture. I'll show you where the choroid plexus is. The choroid plexus is just an area that's concentrated with capillaries that are covered by the ependymal cells. What are the ependymal cells? That's what actually produces the CSF. So there's a choroid plexus in each and every ventricle. When we look at this cross section here, these are the chambers. These are your lateral ventricles. They're lined with ependymal cells. Those ependymal cells are going to produce cerebral spinal fluid. Again, from this view, that's what the ventricles would look like. Here's lateral ventricle 1 and 2. Here's where your third ventricle is going to be. And here's where the fourth ventricle is going to be. So from this view, you can see the central canal, which is here, number 1. And then you could see the median aperture, the second escape route, and the lateral aperture, the third escape route. And if you're more linear, then you can follow the flow of cerebral spinal fluid through here, lateral ventricles, which is 1 and 2. Here's the third ventricle, so it's got to go through the interventricular foramen of Monroe. Here's the fourth ventricle, where it goes through the cerebral aqueduct. And then it can go through the median aperture, the lateral aperture, and the CSF is flowing through the subarachnoid space. So remember, we had the pia mater, arachnoid, and dura mater. There's also under the arachnoid, it's called the subarachnoid space. That's where the CSF is flowing, in that subarachnoid space. And then it's going to get reabsorbed through the arachnoid villi of the dural sinus, which is way, way up at the top, which we see up here. Here's the superior sagittal sinus. Here's the arachnoid villi, this cluster of grapes right here. That's going to filter and reabsorb the CSF. So the arachnoid villi is going to reabsorb that cerebral spinal fluid. And it should reabsorb it and produce it pretty much at a similar rate. Otherwise, you get this condition called hydrocephalus. Now, what can cause blockage? You know, any type of malformation of the bone, any type of birth trauma, if there's a tumor, if there's inflammation, any type of developmental malformation within the brain or in the bones, meningitis, these can all create some sort of blockage within this system. And now things can't flow. So you start to get this condition called hydrocephalus, where the pressure just builds up way too much. But then they create this drainage shunt to get the fluid out of the brain. Okay. They shunt it. These neurosurgeons implant this drainage shunt, shunting the cerebral spinal fluid to the veins of the neck or to the abdomen to get that out of there. Okay. Right here, it's kind of faint, but I'm going to circle that. You can see that orange structure right there. That's pointing to the cerebral aqueduct. Okay. That's your hydrocephalus. Okay, let's take a break here.